So last day, are you able to hear me? My voice?
Yes,
Yes,
sir.
sir.
Yes,
Okay.
sir.
তুমি
Yes,
দেখতে
sir.
পাচ্ছ পেছনে? আমি পেছনটা দেখতে পাচ্ছি না। ঠিক আছে। ওইদিকে গন্ডগোল হলে একটু জানিও, হ্যাঁ? So last day we had talked about uh, certain basics of gene expression. Today we will go a bit ahead of on that. As Ruchi has come in. So today we will go a bit ahead of that, and I will try to explain some things about transcription and related to transcription, and very important thing that is known as promoter. So you know that you have a DNA strand, you have the sequence of amino acids, so the sequence of triplets present, which will code for an mRNA, which will code for each mRNA triplet will code for a unique amino acid. This we have learned, right? So let me put on my video first. Otherwise, my hands movement. So we know that each unique triplet will coding for one amino acid, right? And depending upon the sequence of the triplets which are there, that will be the sequence of the amino acid attached, and that will make the protein or polypeptide. If that be the case, then there is one thing which we must what makes this transcription take place on the DNA. So therefore there must be a region on the DNA which will help some protein factors, say like RNA polymerase, to bind to the DNA and start converting the DNA into messenger RNA. So there must be some sequence in that region, some special region. It cannot bind anywhere just on the DNA. It has to have a particular sequence on which this RNA polymerase will bind, and then eventually you will see transcription mRNA being formed. Translation is later, but first let us talk about what happens during transcription. I'm sharing my screen at the moment. So, is my screen visible?
Yes, sir.
Hmm. So, you have the DNA. You have the DNA from which RNA is made and this protein is present. Now, one thing, this is a DNA exists as a double helix. DNA exists as a double helix. One is interwound. You can see the interwinding over here in this region. It's and when it is wound, RNA polymerase cannot sit and actually do some work, because like two rubber bands are string, you cannot work on an individual strand. So what happens is this needs to be unwound. This is unwound. So a DNA has got one end known as the 5' end. Look at the orange line. One end known as the 5' end and one end known as the 3' end. When replication takes place, it takes place in the direction 5' to 3'. Transcription takes place in the other way. It will take place in the, if this is the 3' direction and this is the 5' direction, this is the 3', this is the 5'. So transcription is taking place on a strand known as antisense strand. This is complementary to a strand known as the coding strand. or non-template strand. So why is it called a template? It is called a template because this 3' to 5', this blue colored strand which you are seeing, this 3' to 5', this blue color strand is saying that is acting as a template on which RNA is going to RNA polymerase is going to bind, and transcription takes place. So you know that the first triplet of any protein is ATG. When RNA is produced, it will just become the opposite way around, AUG. The sense strand is the other way around. এটা যা অ্যান্টিসেন্স আছে তার উল্টো দিকে। It has got the code ATG, GCA, CCT. This is the coding strand. But when it is converted into the antisense strand, it is essentially the other way around. So when you're reading a DNA sequence, normally you will find the coding strand bin, the sense strand given, not the antisense strand given. Now, antisense strand AUG will become ATG will become A's opposite is T. T's opposite is A. It will be TAC, antisense strand. Transcription now will take place. It is making an mRNA. So the RNA polymerase will, what will it do? It will incorporate the nucleotide which is opposite on the antisense strand. So this messenger RNA which is formed, it is the same, it is also in orange color because that has got the sequence as that of the coding strand or the sense strand or the non-template strand. Template, it is called template strand or antisense strand because उल्टो माने बोझाচ্ছে, उल्टा मीनिंग निकल रहा है, therefore it is antisense. Coding, oh, ATG, you can also understand that this is supposed to be methionine. Here the protein will start. But antisense is ulto, TAC. Why? What is that? So, but this antisense strand is the strand on which actually you will have the RNA polymerase binding. Actually, this strand is transcribed, mRNA is made. And this transcript, therefore, ultimately the transcript comes out in the 5' to 3' direction. But the direction in which the RNA polymerase is moving is from this direction to this direction. এদিক থেকে এদিক যাচ্ছে. That is in the 3' to 5' direction. The RNA polymerase is moving. But what it is being made in the 5' to 3' direction. And eventually the protein is made. In transcription, now, there is a region, if this is where all the proteins are starting, here is the where the protein coding sequence is also sometimes known as the ORF, open reading frame, because that's how it's read, open reading frame. So to start the transcription, the RNA polymerase needs to bind somewhere, and that region is present ahead of this. This DNA is ahead of this open reading frame of that protein, and that is known as the promoter region. It is known as the promoter region. Promoter. That region is known as the promoter region. So this is the start point. Start point of transcription. And before the start point of transcription, this region is the promoter region. Now, before I go ahead, let me explain you the structure of a gene and open reading frame. Let me tell it from the eukaryotic perspective. Okay. So what we have first is you have the open reading frame, ORF. Normally in eukaryotes, this is actually right now it's in the form of a DNA. Now before that ORF, actually from here the ATG starts, and here there is something known as stop. So the ORF is over here. So this is just before the stop, the ORF is present, and this is coding for amino acids to form the protein. But there are certain regions even after the ORF which is transcribed. This is known as the, but this will not be translated because it will not form the protein. This is known as the 5' UTR, untranslated region. And you have the 3' UTR. This is also an untranslated region. So where the 5' UTR is starting, this is known as the transcription start site or initiation site. So before the start site, actually, this is now we are talking all in terms of DNA. ঠিক আছে? আমরা DNA নিয়ে কথা বলছি. You actually have this region. This is neither transcribed nor translated. And this region is known as the promoter. And this promoter is the region where different transcription factors, TFs, and RNA polymerase bind. There are different other proteins which also bind to cause transcription. They are known as transcription factors, and RNA polymerase bind to initiate transcription. At around -10 region, there is no zero. No zero. You have the first +1, the transcription start site, and then immediately you have -1. So at around -10 region, there is this -10 box. It is known as the Tribno box. Some people call it the Tata box. Then some other sequence will be something like TA, TA, TA. It can be, there can be minor variations, but it's something like this. This is the region where actually the RNA polymerase binds. This sequence. And you have a region known as -35 region. So this is transcription is not there in your syllabus. I will not go into details of transcription, but since expression control of transcription is there in your syllabus out of strangeness of their brains, we have to study. You have to study this in addition. But you have to know all this, but you don't know transcription. That's how they want it to be. বাজে কি উন্মাদের মতন সিলেবাস বেরিয়েছে গাঁজা না মদ বুঝি না মদ হলে অতটা হবে না আমার ধারণা গাঁজা মদ হলে তাও একটু কিছু তো থাকে আইদার পড়ে যাবে নয়তো কিছু একটা লেভেল থাকে এটা মনে হয় গাঁজা যেটা একেবারে মানে উল্টো পাল্টা চিন্তা করায় So let's get back to this slide. So let us go to the promoter. So you have two consensus sequence elements. Pribno box near -10 with a constant TAAT. This region is ideal for unwinding because it is AT rich. AT means adenine and thymidine. They have two hydrogen bonds in between them. And GC has got 3 hydrogen bonds between them. So this is more tighter than this. So it's easy to unwind. And there is this minus the 35 region with the consistors TTG ACA. The sigma subunit appears to bind here. So the RNA polymerase has got two parts. One is the holoenzyme, the RNA actual RNA polymerase plus a factor known as sigma factor. The sigma factor is present as long as the RNA polymerase is standing on some of the DNA. It helps the DNA to bind. But when transcription starts, the sigma factor is released. Only the RNA polymerase holoenzyme remains. So let us now get back to the place where your syllabus is. Regulation of Lac Operon. So there are three types of gene expression which takes place. One is constitutive gene expression. Whether it be any developmental stage, any sort of stress, a heat, cold, the expression of these genes, they are always constant. Same rate of transcription হয়ে যাচ্ছে বাড়ছেও না কমছেও না করলেও মানে নেগলিজিবল বাড়া কমা মরে গেলে হচ্ছে না বা অসুস্থ হয়ে গেলে হচ্ছে না কিন্তু বাট under other stresses or under developmental condition whether embryo or later stage it remains the same. It is like actin. They are the cell skeleton. Actin, tubulin, vimentin. There are many such cytoskeleton. Like these are GAPDH, glucose glycolysis, GAPDH. Mostly glycolytic. All the glycolytic enzymes they are more or less they are constitutively expressed. Then you have inducible gene expression under heat stress or cold stress. Under cold stress, DREB proteins go high in plants. In animals, you have heat stress, heat shock proteins coming up. Under different developmental conditions, you have different proteins coming up. So under specific conditions, these are expressed. So why one gene is expressed and why one is not expressed, that is all because it's controlling the expression. It's all happening from the promoter region. Then you have a repressible gene expression. Under certain condition, at a particular level, it's being expressed. Then suddenly under a particular condition, it is repressed. So these are the repressible conditions. Now let us look at a lac promoter. এটাকে বড় করি The Lac Operon system. Let us look at the Lac Operon system in details. First, let us look not in details, but can you see this? Those who are online? Yes, no?
Yes, sir.
Yes,
Okay. So, this is where the ORF is starting, or the structural genes. In bacteria, gene 1, gene 2, gene 3, 1, 2, 3. They are one after the other. And then you have a promoter. This is the funny part of it. Then you have a promoter region, right? In bacteria, there is no intron, exon. Let us not bother about ourselves with that. So here you have the promoter. Before the promoter, there is another site here. We are talking about a lactose operon. You have the site known as the operator. And many nucleotides ahead, there is another gene known as regulator gene. This regulator gene, this is the mRNA of the regulator gene. It is closing for a repressor protein. This regulator gene also has its own promoter. Promoter of I. I is the name of the regulator gene. So basically this is an interplay of two genes. This constantly there is this repressor protein is made. When the repressor protein is made, and what happens whereby this repressor protein, it comes and it binds over here to the operator. When the repressor protein binds to the operator, no transcription. When repressor binds to the operator, no transcription takes place. When somehow the repressor is somehow not allowed to bind, when the repressor somehow is not allowed to bind, then transcription takes place, and the genes required for lactose metabolism are expressed into proteins. যে এনজাইম গুলো লাগবে, those enzymes which are needed for lactose metabolism are expressed. This is why one small aspect of it. So when will the enzymes be expressed? When there is lactose in the media. When there is lactose, then the bacteria, this happens in E. coli, then the bacteria needs to metabolize lactose. যখন ল্যাকটোজ নেই, ফালতু জিনটা খরচ করবে কেন? জিন এক্সপ্রেস করে সেই এনার্জি খরচ করে, why should it do it unnecessarily when you don't need lactose is not there. So this is how you look at things like this. Then the next thing which we talk about is let us go into details. So when there is no lactose in the media, the normally lactose থাকে না, this repressor protein is constantly binding to this region, right? So there is no doubt, no problem on this. So now we have talked about the operator and all these things over here. Now let us look into more details of the structure. This is the whole Lac Operon system. This part is the coding region, and this part is the regulatory region. So this is the gene, this blue color I is the gene for the repressor protein. And this is the promoter of the repressor protein. There is a region known as cap site, then the promoter of the Lac genes. Then you have the operator present. And you have three genes. Structural gene for beta galactosidase, that is known as Z, Lac Z. Then you have the structural gene for permease. Then you have the structural gene for transacetylase. Now understand, when the lactose is in the media, when there is this lactose, there is a lactose inducer, it is known as allolactose. So how will the bacteria know that there is lactose in the media? This lactose in the media, some of it gets converted into allolactose. Maybe because of the some residual beta, whatever, whenever there is lactose, some of it, it's an isomer. The structure remains similar. It ulte jaye bond ta. This is lactose, this is allolactose. Now what happens actually, so what you had seen before, I'm trying to bring it, enlarge it once more. This regulatory gene, you had seen the regulatory gene. It is producing its mRNA messenger transcription hoye, then translation hoye protein ta hocche, and this is an active repressor. This repressor goes and binds to the operator site. Operator site is like a block in front. This is the promoter, the promoter region. But after the promoter, there is a region known as O, or operator region. On that, this repressor is binding. When the repressor is binding, no RNA polymerase egote pacche na, roadblock, kadi egote pacche na. It's a roadblock. This you have seen. Now, when lactose absent, repressor is active, operon off. Inducer ta ki boloto? Inducer is allolactose. Lactose media te asle kichuta, some of it is getting converted into allolactose. Now this allolactose, so when there is no lactose, no allolactose, by default eta ekhane chole jacche. Now we are talking that lactose present, repressor inactive, operon on. এই allolactose এসে বাইন্ডিং করেছে, repressor inactive, operator খালি, road clear উঠে গেল, it is open, operator is now accessible to everybody. Now transcription will take place, and what this enzyme, beta galactosidase, permease, transacetylase, all these enzymes needed for lactose metabolism are synthesized. Now there is a thing known as catabolite repression. When there is glucose in the media, the first choice is glucose. Lactose, glucose, everything is regulated, glycolysis, everything is regulated by glucose. Glucose, if I'm getting glucose, why the hell should I go, even if lactose is there? Why should I take lactose? If there is no glucose, there is lactose, then only I need the genes for lactose. So glucose level regulates cyclic AMP levels. Cyclic is ATP gets converted into cyclic AMP. Adenylate cyclase enzymes. So high glucose, what does it do? It represses this metabolism. This is regulated to glycolysis also. And if it's a signal transduction, if there is no cyclic AMP, what will happen? No cyclic AMP means then glucose metabolism will take place. If there is, that means what? ATP gets converted into cyclic AMP. যদি ATP বেশি থাকে তাহলেই only then cyclic AMP will form. This means there is lot of requirement for মানে ATP আছে তাই জন্য cyclic AMP তৈরি হচ্ছে, তার মানে ATP আছে। And if when glucose comes into the equation, high glucose, it will inactivate ATP getting converted into cyclic AMP. ঠিক আছে? Low glucose, no inactivation. Cyclic ATP gets converted to cyclic AMP. Now, lactose present, glucose কম, cyclic মানে glucose কম হলে কি হবে? High cyclic AMP. If there is less glucose, then there is high cyclic AMP. There is this region on the promoter, একটু অন্য কালারে দেওয়া রয়েছে এখানে। That is known as the cap site, cap binding site. It's a catabolite activator protein, this cap. Cap is inactive. Cyclic AMP comes and binds, it becomes an active camp complex. Catabolite activator protein. It's a catabolite activator protein. এদিকে কিন্তু ইনঅ্যাক্টিভ ল্যাক রিপ্রেসার রয়েছে, ল্যাকটোজ প্রেজেন্ট রিপ্রেসার, তাই যে অ্যালোল্যাকটোজ হয়ে গেছে ওটাকে বন্ধ করে রেখেছে। ঠিক আছে, এই সাইডটা খালি আছে। Cyclic AMP scarce. This is not glucose metabolism. গ্লুকোজও কম আছে, হ্যাঁ, গ্লুকোজ কম আছে, আর তোমার ল্যাকটোজ আছে মিডিয়াতে। সো ইট নিড গ্লুকোজ সাফিসিয়েন্ট নেই। গ্লুকোজ কম মিন্স হোয়াট? It means high cyclic AMP levels. High cyclic AMP, active camp. এটা RNA polymerase 6 behind as a double engine. আরো robustly expression, it starts more robust expression of lac mRNA. ঠিক আছে, কেন অ্যাক্টিভ ক্যাপ কমপ্লেক্স হয়ে গেছে, গ্লুকোজ লো। আর এবারে if there is a inactive cap, then double engine নেই। Now think of the situation, lactose present, glucose present, cyclic AMP low, no cyclic AMP. This is inactive cap complex. Double engine হতে পারলো না। Inactive lac repressor আছে, but little lac mRNA synthesized, মানে নিজে RNA polymerase একটু যা পারে করছে। Lactose, but because glucose is there, cyclic AMP নেই। Glucose is there, so there is no cyclic AMP, and the bacteria does not need to synthesize lactose enzymes. Glucose नहीं, it's happy. So इसको बंद करना है। How to बंद? Normally this RNA polymerase, it needs this active cap complex on its back. So when I started, I did not show this cap sites. I told that moment when the repressor binds, this operator binds over here. এখানে যেই bind করছে, there is no transcription block. But even if the block is removed, this RNA polymerase, it has got very less capacity to go ahead on its own. It needs the double engine, inactive lap. So this double engine comes behind and it drives. But when there is glucose, there is double engine or fuel in a. The cyclic AMP is the fuel for the double engine. Clear, everybody? Yes, no?
Yes, sir.
Yes, bolo.
Yes, sir.
Okay. So this is actually Lac Operon. I will pass the note ta diyechi. Then I will send it today. পাঁচ মার্কস এবারে যাও ভাইবা দিতে যাও নাকি এনি কোয়েশ্চেন্স অনলাইন वाले এনি কোয়েশ্চেন্স
No, sir.
নো স্যার ওকে
No.
নো স্যার ওকে আভা বলছে ঠিক আছে then you people go to study now for your practicals কালকে কি অনলাইন করব না অফলাইন করব
Sir, on.
অফলাইন অনলাইন ওকে